Okay, let's just dive right in. Have you ever really stopped to think about the challenge scientists are up against? They're trying to figure out the very building blocks of life, cells. But there's a tiny problem. They are completely, utterly invisible, right? It's a heck of a question because you're not just trying to look at something, you're trying to understand a whole bustling microscopic city. How do you map out its highways, figure out its power grid, or learn its language when the whole thing fits on the tip of a pin? Well, as it turns out, biologists have developed a pretty awesome toolkit over the years. And today, we're going to explore the 5 fundamental techniques they use every day to pull back that curtain and make that invisible world visible. So, first things first. It's kind of obvious, but you can't study what you can't see. This is square one, the most basic hurdle, and the solution is literally the cornerstone of all biology. And that solution is, of course, microscopy. It's a beautifully simple idea. Just use lenses to make things bigger. But, and this is a really big but, not all microscopes are created equal. It's not one size fits all. So let's break this down. Your standard light microscope is your everyday workhorse. It's perfect for watching living cells go about their business. But what if you want to see one specific thing, like DNA? That's when you grab a fluorescence microscope. It uses glowing dyes that latch onto what you're looking for, making it light up like a neon sign. And then there's the big gun, the electron microscope. This thing can magnify things millions of times. That's like being able to see a fly on a skyscraper from another state. So the key takeaway is simple. The question you're asking determines the tool you use. Okay, so, great. Now we can see a cell. But what happens when your experiment needs not one cell, but thousands or even millions of them? You can't just go find them one by one. You need a way to make more. This is the next huge problem to solve. How do you get a big, reliable supply of the exact kind of cells you need to study? Well, the answer is kind of weirdly agricultural. It's called cell culturing, and honestly, the cell farming analogy is perfect. You are literally growing a crop of cells in a dish. You give them all the food and warmth they need to thrive just outside the body. This is a game changer. It's how scientists can test new drugs or even grow new skin for burn victims. And the recipe is pretty straightforward. First, you give them a nutrient-packed soup to eat. Second, you put them in a nice warm incubator. And third, and this is so important, you have to keep everything perfectly clean, totally sterile, because one single stray microbe can wander in and ruin your entire experiment. And about that incubator, it's set to a very specific, very important number, 37 degrees Celsius. Why? Because that's our body temperature. You're essentially tricking the cells. You're making them think they're still inside a person, creating the perfect, cozy little home for them to grow and divide. So, we can see the cells, we can grow a ton of them. But there's another problem. Under a microscope, most cells are basically transparent. It's like trying to examine a clear piece of glass underwater. You know it's there, but good luck seeing any of the details. The solution is just wonderfully simple. Add some color. The technique is called staining, and it uses special dyes that grab onto different parts of the cell. All of a sudden, everything just pops into view in beautiful, vibrant detail. It really is like a biology coloring book. Now, of course, scientists have different stains for different jobs. You have your basic stains, like methylene blue. Their only goal is to make things visible. Bam! The nucleus is now dark blue. Easy. But then you have differential stains, which are a bit smarter. Gram staining, for example, is a classic. It makes some bacteria turn purple and others turn pink. So it's not just showing you that something is there, it's helping you figure out what it is. Okay, we're really getting into the weeds now. We can see the cells, grow them, color them, but what if you need to study their parts individually? How do you get a pile of just mitochondria over here and a tube of just ribosomes over there? I absolutely love this analogy because it's dead on. To separate everything inside a cell, you basically put it all through a super high-speed spin cycle. This technique is called centrifugation. You take your blended up cell soup and you spin it at incredible speeds. Just like when you spin a wet towel, the heavy stuff gets flung out. In the cell, the heaviest parts, like the nucleus, get packed into a pellet at the bottom first. You can then pour off the liquid, spin it even faster, and the next heaviest stuff comes out. Now, centrifugation is amazing for sorting the bigger parts of the cell, the organelles. But what about the really small stuff, like individual molecules of DNA or protein? They're just too light. Spinning them doesn't work. For that, scientists invented a different kind of sorting system. They invented a molecular race. And this race is called electrophoresis. You put your mix of molecules, like different sized pieces of DNA, into a slab of gel. Now, you can think of this gel as a thick, tangled forest. Then you apply an electric current. Since DNA is negatively charged, it starts moving toward the positive charge at the other end. But here's the clever part. The tiny, small pieces of DNA can wiggle through that forest really fast. The big, bulky pieces, they get caught up and move much slower. When it's all done, you have a perfect lineup. All the molecules sorted by size, like runners finishing a race. So we've walked through 5 distinct techniques. But the real power, the true magic of modern biology, happens when you realize these tools don't work alone. They're all part of one integrated, complete toolkit. Let's just quickly recap. Microscopy is for seeing, cell culturing is for growing, staining is for coloring, centrifugation is for sorting the big stuff, and electrophoresis is for sorting the small stuff. Each one solves a very specific, very important problem. And this is where it all comes together. Imagine a scientist trying to understand a specific cancer. They'll start by culturing millions of cancer cells. Then they'll use a centrifuge to isolate just the proteins. Next, they could use electrophoresis to separate those proteins by size. And finally, they might use a fluorescence microscope to see exactly where one of those proteins is located inside the cell. It's by combining these tools that scientists can piece together these incredibly complex puzzles. I mean, think about it. These 5 fundamental tools took us from not even knowing that cells existed to understanding the deepest secrets of genetics and disease. It's just incredible. And it really makes you wonder, doesn't it? If these are the tools that unlock the cell, what will the next generation of tools allow us to discover? What secrets will they reveal for the very first time?